I'd like to welcome everyone to Track 1, Regenerative Therapies. I'm very excited to welcome Dr. Bill Andrews. Dr. Andrews earned his PhD in molecular and population genetics at the University of Georgia in 1981. He has been a medical researcher in biotech since 1981, focusing on cancer, heart disease, and inflammation research, though his passion has always been aging. In the early to mid 1990s, while at Geron Corporation, Bill led the research to discover both the RNA and protein components of the human enzyme called telomerase. Bill was awarded second place as National Inventor of the Year in 1997 for his cancer research. Dr. Andrews was named inventor on over 50 plus US issued patents on telomerase and author of numerous scientific research studies published in peer-reviewed scientific journals. Thank you so much for being with us, Dr. Andrews, and please begin when you're ready.
Thank you very much. So the title of my talk is Gene Therapy and Aging. I have some slides here that A4M asked me to put in. I will not be marketing any products during my presentation. I will be not talking about any companies. I will be just educating people on the field of gene therapy and aging. These are just some of the objectives that you have in your thing. I want to begin with just describing myself a bit so you know where my head is at. As it said in the introduction, I've been working on essentially trying to cure aging for 30 plus years. I've been interested in it most of my life. And why do I spend so much time doing this? And one of the reasons is because there's a lot of great things that are going to happen in the future. For example, in this photo, someday we're actually going to meet life in other planets. And when that happens, I want to be there. I'm also very interested in, I love living. I also love people that love living, and I don't want to see living become any less lovable for anybody just because of something like aging. So I focus a lot on that. And a lot of us take living for granted. It's actually the greatest thing that ever happened to any of us. And I don't think we should just let it get thrown away. So that's why my focus has always been for a long time on trying to deal with the aging process. There's two things. One is aging, and there's also aging related diseases. My talk today will, if I was to talk about all aging related diseases, I essentially be talking about every disease that ever existed, because I believe that 99% of them are age related. So I'm only going to, when I talk about age-related diseases, I'm only going to be talking about treatments whose mechanism of action is related to actual aging. There's a lot of biomarkers of aging. In this clinical studies that I have underway, I am testing practically every one of these biomarkers of aging. But I'm a little concerned about the whole subject of biomarkers of aging because they don't actually mean aging. So when somebody says that they've reversed the biomarker of aging, all they've really done is reversed the biomarker of aging. They haven't actually necessarily reversed aging, so we don't really know. Nevertheless, in my studies, I do all these tests. But what I'm really interested in, I think what most of your patients are interested in, is when they come to you and they say, "Oh, do anything to slow down my aging process," this is what they're talking about. Okay, and I can't really tell you which biomarkers here is telling us. I know that all of us here in this audience know which photo of Betty White here was taken first, but what is the biomarkers we're using to be able to diagnose that? I don't really know. I can put a finger on a few of them, like wrinkles and stuff like that, but it's a lot more complicated than just a bunch of biomarkers of aging. So what patients really want to do is they want to see this process slow down. Whatever that is in those pictures, they want to see that slow down. And then the next question comes in, how do you measure slowing down of aging? To measure the slowing down of aging, you actually have to have like hundreds of identical twins, one receiving a treatment, one not receiving the treatment, and see if after 20 years, one of them looks younger than the other one. And that's a problem too, because usually you have to do such a study with older people, and usually by the time identical twins are old, they're not identical anymore. So it's a really, really difficult thing to do. So in my opinion, one of the really only ways to know that we've actually ever come up with something that actually does affect aging is if it's something potent enough to actually reverse aging, which I call the cure for aging. A lot of people don't like the fact that I say cure, but I am not an expert in semantics. But that is what I've called it my entire life. And so what I'm talking about is the cure for aging. And what I call this the Betty White test. And so even though all those biomarkers that I just showed, if anybody ever tells you that they've got a product that reverses aging because they have reversed one of those biomarkers, I personally will not believe it until they actually can demonstrate that their biomarkers or that their treatment will pass the Betty White test. So that tells a little bit about who I am, but I want to say that I am a biotech researcher. I actually have a biotech company with about 10,000 feet of research space. We've got every piece of equipment you would ever need to actually study aging. And I've had this company for over 20 years right now. By trade, I'm a medical researcher, PhD. I've had a lot of experience. I've been in biotech for 40 years. I've actually been involved in many of the things listed here, beginning with human growth hormone, tissue plasminogen activator, erythropoietin, and on and on. So I've actually had quite a career with a lot of biotech breakthroughs and also a lot of clinical studies in biotech treatments. But in the last 30 years, I've been 100% focused on searching for a cure for aging. My education, who am I to say all this stuff? I've got a PhD in molecular and population genetics, which means I understand the biology of genes and also evolution. Population genetics is mostly the study of evolution. So I understand a lot of things about why we age and what's going on. And that's a subject for another discussion. I could spend a lot of time talking about that. I have a bachelor's in biology and psychology also, but I have a minor in statistical theory, which is something that I consider extremely valuable tool that I have, my ability to be able to analyze other data and be able to analyze my own data and know when data is for real and not for real. I've been in biotech for 38 years, almost 40 years. My focus has been on cancer, inflammation, heart disease, in addition to aging. Mostly the cancer, inflammation, heart disease was before I really started focusing on aging. I was awarded second place for National Inventor of the Year for my cancer research. I'm an ultra marathon runner. I've been featured in 2 documentaries. One is The Immortalist, which is about my research, and the other is called The High, which is about my running, where I ran 138 miles through the Himalayas of northern India at 18,000 feet nonstop when I was over 60 years old. Okay, so I'm also an author of 2 books, Curing Aging and Telomere Lengthening, which I'll discuss later in this talk. So I want to impress on everybody that there's something that I really recommend everybody do, and that's something called meta-analysis of scientific peer-reviewed studies. There's a lot of misleading information out there. A lot of people have heard a lot of things lately, for instance, about hyperbaric chambers curing aging, or they've heard about a cure for progeria, or they've heard about genetic reprogramming curing aging. But all of this is extremely misleading, and most of it is to actually just get publications and get products on the market, or to get investors into companies. So the bottom line is it's not what gets in the press that is the most important. Okay, so do meta-analysis of scientific peer-reviewed studies, which means go to some place like PubMed, pubmed.gov. Read the studies, but don't just read one study, because you can find support for anything that you wish to be true, even in PubMed. Just because some papers get in there all the time that are wrong, you got to read a lot of papers and do what's called a meta-analysis. And later on, I'll be showing a link to a video where I do discuss meta-analyses a lot. So let me begin with results from my meta-analyses that I've done on my own. First thing I want to discuss is what causes aging. There's a lot of theories on why we age, more than I wish to count, but I like to think of them as multiple sticks of dynamite that are burning inside of our cells, and each one's a different cause of aging, or at least a theory on aging, but which stick of dynamite has the shortest fuse? That's really what I wish to focus on first, and then after we put that fuse out, I wish to focus on the next fuse. But what I've learned from my meta-analysis, and actually from research in my own labs for the last several years, including when I was at Geron Corporation, is that humans are very different than mice. Most of the studies on aging in mice don't relate to humans at all. And that's why a lot of drugs work on mice, but don't work on humans at all. So the stick of dynamite in mice, that's the shortest fuse, is different than in humans. There are studies on mice that I'll talk about later, and those mostly involved engineered mice that have been made to age more like humans. So most of the theories on why we age do say that don't say that we're like old trucks sitting in a field. Trucks sitting in a field, they age by the wind, the sun, the rain, and we're not like that. Okay, most of the theories say that we age because our cells age. And so if we zoom in on a human being, we can see that a human is made up of 100 trillion cells. And as I said, most of the theories say we age because these cells age, and that's where we got to target. We got to figure out how to prevent aging in our cells. And one thing that we know for certain is that, and we've known for a long time, that if you grow human cells in a petri dish, they grow at a linear rate. This is at the x-axis is days in culture here, the y-axis is number of cell divisions. And so from day to day, you see that the cells divide at a constant rate. And that's the same for bacteria and fungi and other things too. But in humans, we find that after a while they stop. They don't divide indefinitely. They encounter something called a Hayflick limit, and this was discovered by Leonard Hayflick in 1961. And at the time, everybody thought he was totally wrong, because why would a cell not continue to divide? Well, let's see. I'm getting a message on my computer. Okay, so I'm not certain if that was from A4M or not. Okay, so what the Hayflick found was that when he took cells from a 10-year-old, he found that these cells could divide 55 times before they stopped dividing. Well, when he took cells from a 50-year-old, he found that they could only divide 35 times before they stopped dividing. And when he took cells from a 90-year-old, they could only divide 15 times. Well, what's going on here? How could a cell know how many cell divisions it has left? How can a cell know how old it is? This is something that baffled scientists for a long time. But one of the things the analogy that I thought made the most sense is that cells have something equivalent to ride tickets at an amusement park. Okay, you go to an amusement park, your mother gives you a certain number of tickets, you see how many rides you can go on. You go on a few rides, you see, well, you don't have as many tickets as you did before, but you still have this many rides that you can still go on. So this is happening something like this is a mechanism that could be conceived of as being possible to exist inside of a cell, some kind of counting mechanism, such as ride tickets at an amusement park. So if we zoom in further, let's go find out where these ride tickets are. We find that every cell has a nucleus, and inside the nucleus are found these chromosomes. The chromosomes are where our genes are. They give our hair color, eye color, all of you know that. But the DNA is organized. The DNA that contains all these genes is organized in two arms of the chromosome, the top arm and the bottom arm in this picture. And what you see is that the chromosome is shorter than the DNA can be. The DNA is about 100 million bases in length. They're like beads on a string. The beads are called bases. And so it's all coiled up like a slinky inside of each of these arms. And I want you to think of this long string of beads as a shoelace. And at the tips of your shoelaces, you know, you have aglets that protect your shoelaces from degrading. Well, our DNA has the same thing. They're called telomeres, shown at the tips of our chromosomes. Telomeres are the aglets of our chromosomes. So let's zoom in on one of our telomeres. We find that a telomere is about 15,000 bases in length, at least when we're first conceived. Remember, a chromosome is about 100 million bases. Telomere is a very small thing at the very tip. So 15,000 bases when we are first conceived. And this is where all the troubles begin, because as soon as our cells start to divide, each and every time our cells divide, our telomeres get a little bit shorter. There's so much cell division that occurs between a newborn single cell embryo and a newborn baby that our cells have already shortened down to 10,000 bases before we're even born. Well, the problem doesn't stop there, because we have a lot of cell division left to go. We have a lot of wounds to heal. We have a lot of immune things to fight. We have a lot of things going on growing up. And when our cells get down, telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. That is something that is true for all mammals, or at least all humans. Again, let's go over that. We are conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. And really, till just recently, there's nothing you can really do about this. There's a few things you can do to slow this down, which I'll discuss, and some other things coming up. But the bottom line is that this is a clock of aging, and it's one of the few clocks that's ever been found. A lot of people have found things that uh, do seem to affect aging, such as histone deacetylase uh, factors, let's say genetic reprogramming, sirtuins. These things tend to have very global effects, not on just genes that affect aging, but all genes. And so they tend to have a lot of side effects and do a lot of other things besides aging. So I think telomeres is something important because telomeres in all the research that's been looked at so far seem to target just the genes that are related to aging. So the telomeres are found at the tips of our chromosomes, shown as the ride tickets at an amusement park are found at the very tips of our chromosomes. And this is what we want to work on. I want to say that nothing else, no matter what we do, what else we do to cure aging, nothing else is ever going to work unless we also stop telomere shortening. I said there's a lot of theories on why aging, what causes aging. I'm not saying that telomere shortening is the only cause of aging, but it is the one and only thing that I know of that we absolutely have to do for anything else to work. That is no other theory of aging is this absolute. No matter what we have to do, what else we do to stop the aging process or slow it down, we have to also deal with the telomeres. So I believe that the kingpin of aging is the telomere, and I believe that's true in primates, dogs, cats, horses, sheep, pig, and deer. That's based on meta-analysis. Most animals on the planet, including all rodents, including mice, do not age by telomere shortening, which is a common misconception, and a lot of research has been involved in looking at telomere shortening in mice, but it's always usually been engineered mice. Now, telomeres don't just affect aging, but the best example of where telomeres play a role in aging is this disease called progeria. I had mentioned earlier that there's been recent supposed cure for progeria. It actually it extends the lifespan of these kids about 3 months and gives them terrible side effects from vomiting and stuff like that. It's called something that it's a prenylation inhibitor. Which is something that is actually a failed cancer drug that the inventors of the drug searched for a market, and they came up with a disease called progeria that is also involves prenylation of a protein called lamin A. But lamin A is also published in lots of journals now, articles, to be the cause of the short telomeres that these kids have. So the short telomeres is what's given these kids a life expectancy of about 20 years. And if we could find a way to prevent the telomere shortening, this could be a cure for this disease. And I assure you it would be a lot more than just 3 months extension of lifespan without any side effects. But it's not the only cause. Progeria isn't the only result of short telomeres. There are a lot of short telomere diseases, also called telomeropathies. So just about every disease you've ever heard of now has been published in scientific peer-reviewed journals to be connected to the length of your telomeres. Bottom line is bad things happen when telomeres get short. We have to figure out ways to keep them long. One of the first things that I want to discuss, especially since a lot of patients now are coming to their doctors saying they want to get their telomere lengths measured, is that there really isn't any good method of measuring telomeres yet. These are the commercially available protocols. I'm not mentioning companies here or anything like that. I'm just mentioning the protocols. But they're all very beginning stages. They don't really give you solid good answers. This is an example of one study that's actually pretty typical of most studies, where they do publish data showing that some type of biological process, in this case it's the children born to overweight mothers, that there's a correlation between one thing and another that causes telomeres to show the correlation. So in this case, the mothers, if they're overweight, their children have shorter telomeres. This line is showing that it's significant. But because of my background in statistical theory, I know that statistical significance does not mean there's a strong relationship. It just means that the relationship is maybe a little bit different from totally random. That's all it means. There's a lot of scatter in telomere length measurement data. And actually, when you actually look at the real data, it's not a straight line at all. It's actually a big mess. And so actually to be able to tell your patients that they can get their telomere lengths measured and that this will mean something is actually far from reality. So I believe that we are a long ways to go before we're going to have a good way of measuring telomere lengths. In the meantime, there are other ways to measure aging, biological aging, that actually at this time might even be better. And I'm a telomere expert saying this, but DNA methylation is actually looking really good in scientific peer-reviewed studies, and so is IgG glycosylation. Both these things are good measures if a patient comes to you and says that they want to know something about their biological age. I will tell you that we are at my company developing a new method for measuring telomere length that involves this new technology of long-read DNA sequencing. You've heard that chromosomes are now being sequenced from telomere to telomere, but they're not being sequenced telomere through telomere. And that's because nobody has the ability yet to be able to figure out how to know that they've read all the way to the end of the telomere or if the reaction just terminated early. But we have developed a way to know that we have read all the way through the telomeres, and we're trying to get that in production right now. But in addition to actually measuring the length of the telomeres, we're going to be able to tell which chromosome arm each telomere is from. And this is going to be important because there's nobody who believes that it's all the telomeres that play a role in aging. It's probably one or two telomeres in our chromosomes. We have 23 chromosomes with two telomeres each, so 46 telomeres. Maybe it's only one or two of these telomeres that play any role in the aging process at all. But when we get this new technology set up, we'll be able to identify which chromosomes, and studies will be able to help us identify which telomeres are the ones that we really need to be paying attention to. So as I said, that's in development right now. Big thing is, how do we control telomere length? One thing is we can reduce the rate of telomere shortening already, doing almost everything that our mothers always told us to do when we grew up. One is reduce inflammation and oxidative stress is things that can help reduce the rate of telomere shortening. These are a lot of different things that are things including that you can do that. Exercise, as long as you keep it fun. There is a wrong way to exercise, and that's when you overdo it. Will actually accelerate your telomere shortening and accelerate your aging. But if you keep it fun, it's actually okay. Actually probably decreases your rate of aging. There's three let's say supplements I've mentioned here: antioxidants, omega-3s, vitamin D. For the omega-3s, the studies are now suggesting that 1.3 grams of EPA and 1 gram of DHA per day is the proper amount to reduce the rate of telomere shortening as much as possible. When it comes to vitamin D, 10,000 units per day is probably what you want to do, at least to get the levels of the vitamin D to 60 to 100 nanograms per mil. Antioxidants are important, but of course, too much can cause pro-oxidation. So you want to just do whatever it says on the bottle. But there's a whole bunch of other things here that people can do, like reducing stress. I highly recommend yoga and lose weight and don't smoke, etc. Now, I like to think of aging as a tug of war. And so I use this analogy a lot, but it's pretty much a one-sided tug of war for all of us. Okay, we have a lot of people pulling to shorten our telomeres and also to cause us to age. And there's things that bad things we do, so bad lifestyle choices that can add to this side. And then there's good things we can do to actually remove people from that side, but we can't remove all of them. So in most of our cases, we have what is considered accelerated telomere shortening, because we are doing, nobody lives the perfect lifestyle or has the perfect genetics. But using some of these supplements and lifestyle choices, you can reduce it. You can reduce it down to a basal level, but that still gives us only a theoretical maximum lifespan of 125 years. If you have the perfect genetics and lead the perfect lifestyle, you cannot live longer than 125 years unless we solve this telomere shortening problem. And nobody comes close because nobody lives the perfect lifestyle or has the perfect genetics. Now, one thing that we can do, and that's where we're going to get into some fun stuff, is telomeres actually don't shorten all of the cells of our body. They don't shorten in our reproductive cells. And when I started doing the research on this, and this is important, because if telomeres shorten in our reproductive cells, then our children would be born with shorter telomeres than we have. Their children would be born with shorter telomeres than they have, and would have been extinct as a species years ago. We wouldn't have any chromosomes left. So some reason telomeres don't shorten in our reproductive cells. When you grow reproductive cells in a petri dish, and by reproductive cells, I'm talking about the primordial germ cells, the cells that give rise to our sperm and our eggs. When you grow these cells in a petri dish, they never reach a Hayflick limit. So back in the early 1990s, I was doing, when I got really involved at Geron Corporation to discover why this is, my team ended up discovering this enzyme called human telomerase. The green squiggly thing here is showing the telomere, shown as a double helix. And the factory looking thing is telomerase. It binds the very end of your chromosomes and lengthens telomeres. The G's and the A's and the C's are bases that are being added to the telomeres to re-lengthen them after they shorten. Now, telomerase is actually, as I mentioned, not all animals age by telomere shortening. Some animals have undetectable aging. That includes, the first one to be discovered was lobsters. It was published in back in 1998 that lobsters seem to have no aging process, and it's due to ubiquitous telomerase expression, meaning that they have telomerase produced in all of their cells. They have no telomere shortening. And there's a lot of people that say, well, telomerase causes cancer. I'm going to come back to that later. But these actually lobsters, they rarely get cancer and other diseases. The same is true for clams, tortoises, humpback whales, some fish, and some birds. There's actually a tortoise. Charles Darwin had a pet tortoise named Harriet. She only died recently at 180 years old. There's whales that have been found to be 150 plus years old by different markers, including finding old harpoons in them that carbon dated show that they were 130 years old. The only animal, though, shown here is that has anything that's equivalent to rings on a tree that we can count are actually clams. And if you look at these clams, you can see they have stripes on them. Well, they get stripes, new stripes every year. And so you can count the number of stripes and figure out how old they are. And lo and behold, they have found clams that are over 500 years old. All of these animals have no telomere shortening. They have telomerase produced in all their cells. They rarely ever get any kind of diseases, and they have no detectable aging. And the only thing really mainly different about them in terms of aging that between us and these animals is that they have telomerase produced in all their cells, and they're not suffering any side effects that are really bad. So I essentially would like to see us all be more like the lobsters. Now, there are approaches to produce telomerase. Before, what I had mentioned was things that could prevent decreased telomere shortening, but there are things that we can do to produce telomerase that could actually help lengthen the telomeres. These come into three categories: nutraceuticals, pharmaceuticals, and gene therapy. But in most cases, all they're really doing is adding maybe one person to the other side of the tug of war. We still lose that tug of war in most cases, but we do slow it down a bit, which is by using anything that produces telomerase in our cells is one way that we could possibly extend our lifespans beyond 125 years, lifespans and health spans. So let's talk about now gene therapy and aging, which is the main topic of my discussion. Gene therapy, also called gene delivery. I want you to think of a human cell as a soap bubble, and think of another soap bubble that's a little smaller. That's the gene therapy. And inside that gene therapy, that small soap bubble is the gene or mRNA for human telomerase in the case of telomerase gene therapy, but other gene therapies will have other genes in there. And we all know that when two soap bubbles come together, they fuse. And when that happens, the gene therapy gets inserted into the cell, and that's as simple as it is. Okay, when the gene gets inside the cell, it starts to produce telomerase, or if you have another gene, it produces whatever that is. But in the case of the gene therapy, you can engineer these genes to get them turned on dramatically so that they actually, in this case, produce lots of telomerase. And so this gene therapy protocol has been used since 1998 when we first discovered telomerase. We discovered it before 1998, but when we did a study that was published showing that we could actually extend live cells through the Hayflick limit. This is again showing pictures of cells dividing in a petri dish. Normally they reach the Hayflick limit, but when we produced telomerase inside the cells by gene therapy, telomerase gene therapy, we found that we completely obliterated this Hayflick limit. It's the first time in the history of the planet that anybody had ever done that before. We published that paper. It was thought by everybody that it was impossible to prevent what's called senescence, which is the Hayflick limit. So a lot of other studies have been done. People have taken now, people that actually, after I left Huron Corporation to start my own company, people remaining there did experiments where they put human skin on the back of mice. They they had young skin and old skin. They also put telomerase in by gene therapy in some skin and some without. And they found that when they did telomerase gene therapy of old human skin grown on the back of the mouse, it looked visibly younger and it behaved young. And when you looked at the biomarkers of aging, which as previously said, I don't necessarily believe in, but they did show a dramatic reversal of the biomarkers of 30 different biomarkers that were looked at. And then there was also a more recent study where Dr. Ron DePino actually did, using engineered mice that did age by telomere shortening, was able to show that by lengthening the telomeres, they were able to reverse aging in practically every way imaginable. In that study, they showed a 33% increase in telomere length, which is the first thing you got to show before you can ask anything else. You got to show that you actually did increase the telomere length. But they showed restoration of fertility, restoration of spleen size, sense of smell. And what was most exciting is they saw restoration of brain size and function, which is one of the first studies ever to suggest that maybe dementia is not the loss of memory, but loss of access to the memory. Just recently, there's been a new study that came out with something called integrated stress response inhibitor, where they're actually finding the same thing. They're saying that this is the first time anybody's ever shown that loss of memory might actually not be loss of memory, just loss of access to the memory. But we actually showed this over 10 years ago, maybe 20 years ago now. But gene therapy is the only thing so far that can actually put enough people on the right hand of the side to actually lengthen telomeres. And this is what's getting excited. This has not been tested in humans yet, but before I get into that kind of stuff, I want to talk about I intentionally put a lot more slides into this presentation that I can possibly show, but I want to provide you with references where you can go and read yourself if you're interested in following up on this. There's been a lot of gene therapy studies now done in animals, mostly mice. There's also a lot of different things besides telomerase have been used in treating aging in mice and other animals. In most of the gene therapies, there's been 4 different types of vectors used. One is called lentivirus. The problem with lentivirus, lentivirus is derived from HIV. The problem with lentivirus is it integrates into the chromosome, and when you integrate into the chromosome, this can cause mutations that lead to cancer. So the virus itself causes the cancer, not what the gene is, not what the virus is delivering. Adenovirus doesn't integrate in the chromosome, but it does have a tremendous number of immune responses that it causes. So a lot of people suffer from cytotoxic T cell responses, et cetera, when treated with an adenovirus. Adeno-associated virus, essentially the new kid on the block, doesn't have these first 2 problems, but it does have a very low infectivity rate. We find that when we infect cells here in our lab in a petri dish, we have to use essentially 30,000 times more virus than we do when we have cells in order to get at least 50% of the cells infected. So the infectivity rate is very, very low for the adeno-associated virus. Non-viral vectors also exist, but their infectivity or their delivery rate is even lower. So, but the best kid on the block right now is adeno-associated virus, and that's what we're doing. But we do need a new vector. That's something that my company is also working on, trying to come up with new alternatives to come up with some delivery system even better than AAV. But using AAV, there's now been 250 clinical studies used that have studied this. There have been no serious side effects except there's been some little bit of immune sensitivity. 70% of the people that get treated already have antibodies or other neutralizing substances in their blood that actually prevent the gene therapy from working. That's why I say if you treat somebody with a gene therapy, you got to verify that it lengthens telomeres before you can say anything else. They found that the DNA in the virus is actually immunogenic itself, so that cells that get infected are actually, the immune system attacks them. And then a lot of people can't be retreated because they've already developed an immune response against the gene therapy. So these are problems with it, but there's been no actually serious side effects that can't be dealt with. So these problems here are considered minor adverse effects. FDA has provided some new rulings that have made doing gene therapy a lot simpler. One is that I'm just let you read this. They've allowed for now rapid approval of approval of being able to do clinical studies of life-threatening diseases that can be treated by gene therapy and other kind of approaches. And there's also this other new thing that is allowing some of these genes are extremely expensive studies we're doing us $1 million to produce to treat one person once. Okay, so these are extremely expensive clinical studies. I'm not saying this is ever going to be a treatment that people are going to buy in the future. Maybe a few will, but I'm looking at this as a great proof of concept for showing that telomerase lengthens telomeres and treats aging. But ultimately, hopefully, there'll be a cheap pill that we can all take to do the same thing. But gene therapy, I'm getting another message from probably A4M, but gene therapy it now has been shown that the government has now allowed the patients to actually pay to be in the study, which will allow some of the studies going on to be done where they normally couldn't afford to do the studies. There are a few gene therapy studies underway right now. I'd mentioned other things that have been done to treat aging, but they're not in clinical studies. But there's 3 clinical studies: one for telomerase gene therapy, one for clotho, or a few for clotho. Let's say a few for telomerase, a few for clotho, and a few for follistatin. So keep an eye on those. There's a lot of age-related disease targets. I agree with the FDA. You can't target aging as a disease because it's not measurable. I showed you the Betty White's test. That's not a real measurable test. That's pretty much just the way we feel about things. But there are a lot of different age-related diseases that are measurable. And most at the top of this list, age-related dementia is what we are focusing on mostly. So I want to talk about treating age-related dementia with telomerase gene therapy. I'm going to go fast through these slides. I'm not keeping track of time, but I assume I'm running short on time. You can read these studies. There's been a lot of studies done to show that telomere shortening causes brain decline in mice. These include a decreased so the telomere shortening, not treating the telomere shortening, causes decreased cognition, decreased neurogenesis, on a lot of different things I'm not going to go over. You can read these in your slides, that's your handouts. Now, the telomere lengthening now has been shown in several studies to actually reverse these things, and they've shown these ones in particular. The telomere shortening has been shown to correlate with brain decline in humans, even though nobody's actually done anything to actually do a study on humans yet, a treatment study. Telomere shortening does correlate with brain decline. Here's a lot of studies. You can follow up on those. And then, oh, this is one more study that I thought was a recent study showing that telomere shortening does affect Alzheimer's disease. And usually when I show this red box, this is text out of the paper that focuses on that. It's in your handouts. One very exciting paper that just came out recently, Parkinson's disease has now been shown, I think, in a very good study to be shown to be correlated or associated with telomere lengthening. There was a few studies beforehand that argued that maybe it doesn't, but now I think this study here actually settled that decision. It does. So their findings that leukocyte telomere length is associated with Parkinson's disease. Okay, so I want to address a few issues with clinical studies that we have to study. One is AAV gene therapy safe to the patient? Is it safe for everyone around the patient? And is telomerase safe? And again, I'm just going to let you read these papers. These are papers showing that telomerase gene therapy is safe. Or that these papers here are that telomerase, that adeno associated virus is safe in a lot of different studies. There's not a single study that has shown anything except for some minor immune response problems. Then is it safe for everyone around the patient? Again, there's a lot of papers showing that people around the patient are not going to be infected by this gene therapy, or nothing's going to be transmitted to them in various levels. Again, the red boxes will highlight what I've read in the papers. Then is telomerase safe? And again, there's a lot, of course, telomerase hasn't been used on humans yet, but it has been shown to be safe in mice, and a lot of various approaches. Again, here's papers just showing this safe in mice. And then one of the big concerns, and I mentioned this earlier, is that a lot of people believe that telomerase causes cancer. As I'm going to mention in a little while, I'm probably one of the reasons why a lot of people think that is, and I'll explain why that is in a second. But there is no clinical data whatsoever saying that telomerase causes cancer. A lot of people think that you put telomerase into a cell, it turns them into cancer cells. There's not a single piece of data that's ever shown that that happens. We do it all the time here at my company. We've never seen that happen. There's been a lot of efforts to show that it caused cancer, including the study where I mentioned before, where they treated engineered mice with telomerase. They were actually trying to show that these mice got cancer, and they actually were surprised to find that it actually reversed their aging. So for every study that suggests that telomerase might cause cancer, there's been at least 10 studies that show that it's actually the lack of telomerase that causes cancer. Telomeres, so telomeres cause, when telomeres get short, they cause mutations and chromotherapy. This is why some people say short telomeres cause instability in chromosomes. The mutations in the chromotherapy is just when telomeres get really short, you can see in the microscope all kinds of rearrangements, which are rearrangements of the chromosomes, which are mutations. But these cause cancer. They cause telomeres to not shorten. Some of the mutations actually cause telomerase to turn on. They cause metastasis, and they cause cancer to survive whatever you hit them with. If you treat them with chemotherapy, the cancer goes away, but the mutation rate's so high that some of these cells develop a mutation to survive. So I believe you want to keep the telomeres long when treating a person with cancer. Again, here's some more papers saying telomerase does not cause cancer. Telomerase decreases the risk of cancer, and there's two places where you can go. One is a book I've written called Telomere Lengthening on pages 64 and 77. I go into a lot of detail there. Then in a video that's on my website at sierrasci.com, beginning at 2525, you can see where I've gone to an excruciating detail to explain why telomerase does not cause cancer and why everybody who says so is mistaken, what they're misinterpreting. Okay, so those are two places where you can go if you really want to address the question of does telomerase cause cancer. Bottom line is, when it comes to a clinical study, you got to ask yourself, when is the risk from no treatment greater than the risk from treatment? And so there's a lot of diseases, including Alzheimer's, another form of dementia. They're 99% of the time, or at least in Alzheimer's, always fatal. And so there's a disease where the patient's probably better getting treated with something to try to treat the Alzheimer's than not getting treated, even if there's risk in the treatment. Aging is another high risk disease. In fact, you know that if you're a healthy 89-year-old, you have an 11% chance of dying by the time you're 90 in just 1 year. So already just being old is a high risk. And so you want to do something if the risk you feel is lower than the risk of, if you feel the treatment is lower risk than not being treated, then I would say it's time to do it. The most serious risk to the patient is sometimes not treating the patient. Okay, there's critical issues in clinical studies I alluded to before. You can't just treat somebody with something and then ask, do they get younger, or does it improve their age, help their age-related diseases? You got to first find out, did the gene therapy survive in the blood? And to do that, you got to, how do you measure it? How to make the gene therapy to survive? You got to figure out ways to do that. Does the gene therapy even enter the cells if you get it, if you infect a person with it? And so gene therapies are viruses. I don't usually say that, but you do infect people with the gene therapies, and you got to show that they enter the cells. How many of the cells do they enter, and again, how do you measure it? As I mentioned, the AAV infects very few cells. You have to use high doses to actually get infection. Once it gets in the cell, does it actually produce the gene? In this case, hTERT, which is the protein component of telomerase. Again, how do you measure it? Does active telomerase get produced? You got to, how do you measure it? You got to do all these things. Do telomeres get longer? And this is really important. So you go through all the process. You got to ask, do the telomeres actually get longer? And how do you measure that? And then if they do get measured, then and only then can you ask, does telomere lengthening provide benefits? And again, how do you measure? And is there any side effects, adverse effects from the gene therapy, from telomerase, from lengthening telomeres? And if so, how do we prevent them? So there's a lot of, this is one of the reasons why clinical studies are so expensive. If you do it right, it's going to cost a lot to do. There's a lot of doctors involved, a lot of testing involved, a lot of hospital costs, and then even the production of the gene therapy is extremely expensive. I'm going to close now with recommending 2 books that I've written on aging in general. Telomere lengthening and curing aging. And I'm just, as you can already guess, I'm on a mission to cure aging. That's what I'm doing. I hope to see you there. Thank you very much.